get started. So, there actually is a little reason to the madness here of bringing two groups together. We have a new vet med program that is modeled after the highly successful WIMU program that just started this year. And when we put that program together, one of the justifications for it was to be pursuing this, it's kind of a buzzword right now, One Health aspect of medical training. And that when you're really talking about health issues, and obviously the context of immunology, it's going to be responses to infectious diseases. There's a real advantage to having a perspective of impacts of infectious disease, immune responses, not only in us, but with animals, and I'll get to the reason for that and so forth. We only have, 5 vet students? Raise your hands. So, right here. So they're small. Currently it'll double in number by the end of the week. Hopefully we have all the desks in. I made sure that Mark took blame for that. That wasn't me. I was sending reminders months ago that we wanted to get this room set up so it'd be a little more comfortable and more interactive. I have set up on the canvas for the WIMU students, and then we're doing it currently manually, sending it by email to the vet students. My first 3 lectures, did everybody see those? Okay. And then I also had the syllabi, where I've laid out what I'm trying to get through in this course. What I will be doing, we will get as a resource practice questions. This particular course on the WIMU side historically has been somewhat of a nightmare, I think, from the student's perspective on the exam side of the equation. When I say nightmare, was that we changed the common final every single year, from beginning to the end. We rewrote the common final every single year. And we rotated the writing of that common final of who took the lead between the different sites. So, that's a very variable right there. I write a question totally different than somebody in Seattle or maybe Alaska and so forth. We went away from that a couple years ago and started to use old questions from the board exams. And then that became a security issue, and that we certainly couldn't give you examples of those questions. That's the most secure vault that I've ever had to deal with on the computer. And the internet is to gain access to those. You get this secret code. Mark would have to come to my office, give me the secret code. I'd get on, I'd be able to see the questions and so forth. And we did that in the last couple of years. Now we've gone back this year to kind of a mixture between that approach. You're going to be getting questions in that style. The quizzes that you're going to get will definitely be in that style, and so is the common final. But the common final actually was written by the instructors at the different sites this year. You got to get a 70% on that common final, and that's the real target. And I spent time going over examples in the past. I leave a lot of time at the end of the course for review and so forth, and I urge you to take advantage of it. The one thing that I've told classes every year when I started, that I made a huge mistake in the second year I taught this course. So I've been teaching the WIMU side, I think, since 2000 or something like that. And I gave a copy of a previous common final to the students for practice and for studying. They went in and they took the common final on their own, and they aced it, and thought, oh, I know immunology, and they didn't study. It was the only class that didn't come out either number 1 or number 2 of all the sites. And it's because the common final that I gave out was a little too easy. So what I've done now is I've gone into multiple common finals in the past, and I pull out examples of the different styles of questions, and you guys will get all that. On the vet side, you guys already have examples of questions that should be already sent out. Those are all coming from WSU. We don't have the issue of the different sites writing the exam and so forth in that context. Now, we are doing immunology, which really should take a year, in my view, to do properly if we're going to combine it in the context of infectious disease. And we're going to do this in a month for the WAMI students, and then the vet students get 2 months on this. And why? Well, think about, so the WAMI students, you're dealing with one animal. Vet students now, you're going to be dealing with multiple animals, and we do have differences in the context of the immune system and so forth. So, but there are common aspects of our immune system that we've actually learned from animals. It's what's guiding us in the human, and actually it's what's guiding the use of new classes of drugs in the human. And there are basic mechanisms that are common, and we're going to spend a little over 2 weeks, is it 2 weeks? Yeah. Covering those. And I'll pull out examples of relevance from the human clinical situation, but also the veterinary clinical situation. A lot of times just mainly to drive home that the stuff I'm talking about is real. One of the knocks against immunology is its phenomenology, that we have a lot of immune mechanisms, we've got a lot of acronyms, we got a lot of names and so forth. How much of it really can you use as a clinician with your patient and so forth? The cool thing about immunology now, you can use a lot of it. We have a number of new drugs that are in the clinic that are targeting immune mechanisms. So, this by design wasn't on your PowerPoints that I gave you. Anybody know what this is? Yeah, I just found out this morning, it's the most downloaded picture of Ebola. And so I figured, okay, right, I'm going to use, one of the most hottest bugs out there as an example of the importance of understanding immune responses, not only in us, but in animals. Animals are reservoir for a number of infectious diseases that you may have to contend with as an MD in the clinic, and certainly for vets dealing with animals, that can cross between species. And so Ebola has a reservoir within animals. Current data kind of suggests that it's in bats. I originally thought that maybe it was in monkeys and so forth, but it's the current data looks like it's in bats. And then there will be a transfer to the human population. And depending upon the host that the bug is in, we get different pathology and so forth. We've got many, many examples of this. Emerging and re-emerging diseases, something like 60 or 70% come from animals that make it into the human population. There's other examples on the virus side. You've heard of West Nile, equine encephalitis virus. These all cause significant pathology in humans, but obviously in animals as well. Outer back door, brucella, brucellosis in the bark bison and cattle, that can get into us. In fact, you read about bison all the time. What amazes me, brucella is considered a select agent. And what that means is it's tightly regulated by the CDC because it can be used as a weapon of bioterror. In order to work with brucella, which we do out in our facility, we have to have an extremely high containment laboratory that controls all airflow, everything gets filtered. You have to have approvals from the CDC in order to work with this. And here we have the bug out our back door down at the park, and bison that are coming out and so forth. And it's also in our elk. And any hunters in here, something like 10-20% of the elk that we're harvesting can have brucella. And so at that point, we're potentially exposed to a select agent. you as a clinician having a decent understanding of the background of where the reservoir of these bugs can be can help you in the diagnosis. If a patient comes in and presents with, and here's the problem, the initial symptoms of these patients are very similar. We have to have protocols in our BSL, it's called BSL-3 lab out here, to anytime somebody comes down with something that might be indicative that they got exposed to the brucella they're working with out there, or the Coxiella, or the Yersinia, or the Francisella, they all present the same way. Fever, flu-like symptoms, and so forth. And so they can come in and they may just look like the common cold to you, or a flu symptom and so forth. But if the person's out working with animals and just recently shot an elk and harvested an elk and came in with these symptoms and so forth, it's something to look at, whether or not they potentially have brucella. Very important, because if you're going to treat that patient, you need to start them on antibiotics really, really early in the process. So what am I doing with this course in order to get through it in the time frame that we have? In today's lecture, since I'm assuming we don't have an equal level across the board of understanding of the immune system, I'm going to give a quick overview of the key features that we're going to talk about in detail. So that's the goal of today's lecture. Then what we used to do in the previous 5 years was to talk about innate immunity and inflammation, and we actually gave pathology lectures at this point. And it worked to a certain extent, but we really got into disease processes in those lectures. And then I would come back and go back to the molecular mechanisms involved within the immune system. This time we're going to do it the way that our core molecular mechanisms involved in the immune system. And then we're going to do our inflammation pathology lectures as we lead into when we split into human disease-specific aspects and veterinary disease-specific aspects. So on the surface, it seems like this should work, but we'll see how it unfolds for you. So today, it's going to be a quick overview, and then tomorrow we're going to get into the nuts and bolts of how we generate an antibody. And most students in here have actually heard me lecture before, and I've talked about antibodies and kind of forewarned people that I'd be talking about the genetics behind our antibody repertoire. We'll spend a lot of time tomorrow on that. So we're going to do this quick overview. I always start off this particular lecture with this slide, is that understanding these mechanisms of the immune system that we're going to go through leads to incredibly powerful drugs. In fact, our best success story in human medicine, in my view, is the use of vaccines. And in fact, we have eliminated infectious diseases from the face of the earth solely due to the use of vaccines. And smallpox is our pet example that we always use. And I'm going to be coming back to the smallpox story a little later in today's presentation. So, our immune system is what makes it so that we can deal with pathogens. You guys are going to learn a lot about pathogens and subsequent courses for the WIMU students. It'll be in one single microbiology course. For the vet students, you get courses focused on viruses, you get a course focused on bacteria, and so forth. So we have different types of bugs out there that can cause disease. And we have different immune mechanisms that can target these different organisms. And we're going to be focusing on those. If the bug exists outside our cells, then we're going to spend a lot of time talking about antibodies. Antibodies protect our extracellular spaces. If the bug exists inside cells, antibodies have a tough time getting to them, though some do, but it's not really our effective mechanism for getting rid of bugs that exist inside our cells. We have cells, cytolytic cells, that can target and kill that host cell that the bug resides in, and then exposing it to antibodies and so forth. So the nature of the bug will dictate the type of immune response that'll take place and lead to hopefully elimination. So we'll have some definitions. So a pathogen, just simply an organism that can cause disease. So we have classic pathogens, Ebola, that we started off today's lecture with. But we also have opportunistic pathogens. So an opportunistic pathogen could be a bug that doesn't routinely cause disease, but when you're immunocompromised due to some reason, then the bug can replicate, cause disease to a certain extent, and we refer to those as opportunistic pathogens. You'll have to deal with both in your careers. Classic pathogens and opportunistic pathogens. They can be bacteria, virus, fungi, protozoans. And actually in my previous slide, I threw in prions, so it's both the infectious proteins that you may have to deal with sometime in your career. Infection is simply the presence or replication of an organism within a host, resulting in disease or the potential to cause disease. Virulence is kind of a term that we use back and forth with pathogenic, but it's a measure of the ability of an organism to cause disease. And we have examples. Variola is a virulent pathogen. It's also considered to be highly pathogenic, and so you're going to see these terms used interchangeably, virulent or pathogenic. And again, we'll come back to these examples of these viruses. Vaccinia virus is not very virulent. It doesn't cause overt disease, and it's not very pathogenic. These two viruses are actually similar. This one causes a bad disease, smallpox. This one causes cowpox. And we're going to come back to this story in a few minutes. So, as you guys know, bugs have been the scourge of both veterinary medicine and human medicine for a long time, and we've had multiple examples of incredible impacts by infectious disease. So the Black Death, or the plague, which is due to a bacteria called Yersinia pestis, that again, you're going to learn a lot of details about from in your subsequent courses. It killed a quarter of Europe's population in the Middle Ages. You guys have probably read about the flu, the 1918 Spanish flu. Numbers vary, but anywhere from 20 to 50 million people were thought to be killed by that infectious agent. So how do we fight off these bugs? So we've got to deal with parasites, viruses, bacteria, fungi. And I've just given a couple examples here. Toxoplasma is a parasite. Aspergillus is a fungi. Brucella, the disease brucellosis, is a bacteria. And then flu is a classic example of a virus. We started off with Ebola. And all of you are going to become intimately familiar with the influenza virus. And we'll spend time talking about this particular virus and why it's a problem each year within the human population, and why animals are right at the cornerstone of the potential problem with this particular virus. So if anybody's reading even the popular press and so forth, you might be familiar with this classification of influenza viruses. H is for hemagglutinin, N is for neuraminidase, and these are some of the antigens, the molecules that make up the virus, that depending upon changes within those nucleic acid sequences, they'll give them different numbers. So H3N2 is just one that's different than H5N8. H3N2 is the one that currently, I read last week, was just declared an epidemic in the US. And the yearly seasonal flu vaccine is not effective. It doesn't look to be effective against this isolate of influenza. This guy, H5N8, anybody know this particular isolate, what its big problem is? This is the bird flu. So if you're reading the literature or the popular press last summer, that in Europe, chicken and turkey populations had to be wiped out. This is a form of the influenza virus that currently is mainly restricted to birds, but when it does make it to us, it's incredibly pathogenic. This one really isn't. You know, it's causing problems, but the percentage of infected people that might succumb and potentially even die from the virus is far, far less than the bird flu. The bird flu, highly pathogenic, but has a low level of infectivity for humans. The problem with these particular viruses when they exist in animals is that when the animal gets infected by different influenza viruses, and those viruses get into the same host cell, they can exchange genetic material. And so the concern with the bird flu, H5N8, is that at some point it will acquire a greater infectivity. Currently, it's low for humans, but when it does get into humans, it's highly, highly pathogenic. The big scare is that at some point, and unfortunately, I think this is inevitable, at some point there will be a recombination and there will be increased infectivity. That's just the prediction for influenza. It's the reason why it receives so much attention in vaccine development and so forth.
Can I do that?
Yep.
Can you explain the high pathogenic but low infected?
So pathogenic is the ability to cause disease, and so leading to organ failure or necrosis or whatever, that'll be a symptom in your patient. Infectivity is just simply the ability to get into the host. And so the bird flu has a poor ability to attach to our host cells. And so it has a low infectivity, but if it can attach and gets into our host cells, the pathology it's going to cause is very high. So infectivity is just the ability to infect, and then virulence or pathogenicity is the ability to cause disease once it infects. The ultimate bug, well, it's not really the ultimate bug. So Ebola has high infectivity if you're exposed to fluid, right? Highly pathogenic. Now, that's not a successful pathogen, right? Because it's killing its host. That really doesn't define success in that context. Where it's successful, it's when it's in its reservoir and is able to maintain its population and so forth. But it gets into us, it's highly pathogenic, highly infective if you're exposed to the fluid. All right, vaccines. That's what I started off my talk with. Give a little bit of historical perspective on vaccination and our smallpox story that we already mentioned. This is caused by the variola virus. In the absence of treatment, it still has a 20 to 50% mortality. And our antiviral drugs, though they can be beneficial, do not provide an absolute cure for somebody who is infected by variola that isn't vaccinated. This was an incredible problem, led to, you guys know all the stories, so I'm kind of preaching to the choir here, in the context of the impact of smallpox and this viral infection worldwide, particularly in the Americas, when the Europeans came and introduced smallpox into a population that had never seen it before, and now it had no underlying baseline immunity and so forth. The disease is devastating in populations. And in fact, in some of the data from following 1492 and the introduction of smallpox into Central America and so forth, if you believe anybody read this book, 1492, it's kind of a cool book to read as a scientist because it's referenced all the way through any statement in it and so forth. And he does a back calculation on the impact of smallpox on some of the communities in the Yucatan and other areas based upon kill rate and so forth. And it looked like, you know, there could have been a 90% death rate in some of the cities and so forth due to smallpox. At the time, there was just no underlying baseline immunity against this virus, and it was very, very devastating. And then when he took those numbers, went back, he actually proposes in this book, there was 120 million people existing in the Americas at the time, and it's the impact of smallpox on those populations. You had a question?
Yeah, I was wondering how we define the fact that it's eradicated. What numbers allow us to say that the disease is eradicated?
You know, based upon that, I've got that figure again, and I'll come back to that. We can talk about it. Actually, it would be a hand-waving answer that I'll give you. It's over a period of time that no new cases will be identified, but what that length of time is, I would predict would vary between disease-causing agents and so forth. And again, I'll come back. That's a good question. So, our first vaccine, it was generated by Jenner, and he noticed that dairy maids that had developed cowpox lesions at the time did not seem to get smallpox. And so cowpox is caused by a virus that's very similar but different than the variola virus, and it leads to, in somebody who gets infected, these pox or pustules, sites of viral replication, but not systemic disease, and it's normally self-limiting. It's caused by, like I said, a similar virus. It's called vaccinia virus, and the disease in human, like I said, is mild. And what Jenner did was to take scrapings of some of those pox, those lesions, and gave it to a young boy, and then exposed the young boy to infected individuals, and he was protected against smallpox. Now, obviously, this was not an ethical approach to go in and show this, but what we now know was the cowpox virus is similar enough to the smallpox virus that when our immune system targets it for elimination, some of the mechanisms that are generated against cowpox will work against the smallpox virus. And so people that are exposed to cowpox generate an immune response against the virus, and some of it is cross-protective to when they're subsequently exposed to smallpox, they have a level of immunity that prevents disease. Now, it's not 100% effective, but it was our first example that we can teach the immune system how to fight off a future pathogen without ever being exposed to that pathogen. That's really the lesson that came from this. Yeah.
How did they know that it protected them? It just wasn't something that happened?
Yeah, just leap of faith. You know, this is the story we give, and at the time they wouldn't have had a clue. And it's an N of 1, right? That's the story, but I would suspect that he probably did this with a lot of kids at the time and so forth. Because you couldn't make a conclusion off an N of 1 as well in that context. But this did start the whole field of vaccinology, development of vaccines to teach the immune system how to fight off a pathogen prior to us ever being exposed to that pathogen. And so the term actually comes from gow, and it was this cowpox virus, and it was seeing what was taking place with the human that interacted with animals and so forth that led to this whole field of vaccinology and so forth. So back to the questions. Here's my figure again. if this figure is correct, if the last detectable case of natural infection was in 76, then it was 4 or 5 years later that they declared it officially eradicated. I don't know if that's a standard period of time or not. I think you would actually want to wait longer than 4 or 5 years in that context. But if this figure is correct, then that's where we're at in that time frame. But smallpox is still a problem today. Not naturally, but both the US and Russia weaponized it. And so it is a concern on the biowarfare side of the equation. You know, you guys didn't get vaccinated to smallpox. I did. And actually, it's kind of amazing. That immunity is really, really long lasting with the early smallpox vaccine. And it'll be interesting to see where this goes. You know, post 9/11, there was a lot of talk of restarting, you know, global vaccination protocols and so forth. And smallpox was on that list. I don't know if we'll go that path, but there was tremendous concern since it was weaponized that you know, smallpox could be used and maybe it would be worthwhile to pull the vaccine out and so forth. Yeah.
When they vaccinated the babies, did they get the little droplets?
Oh yeah, they wouldn't have gotten the natural disease. Yep. All right, so what we learned from those experiments that were done is that we can teach the immune system. And that's really the focus of my lectures, is that our immune system can be taught how to fight off pathogens. And that's the whole concept of a vaccine. It is purely used to teach the immune system. And I think I've said this multiple times in previous courses, maybe already today, it's our biggest success story in human medicine, is the use of vaccines. And a single individual is credited with 8 of the 14 childhood vaccines and is credited with saving more people on this earth than any other individual, and it's Montana. So Murray Silliman, who grew up in Miles City, went to MSU, went to microbiology, though it wasn't called microbiology at the time, actually invented 8 of those 14 childhood vaccines, forms of which you guys will all use in the clinic. So how do we fight off pathogens and so forth? We've got different mechanisms, but we actually have 2 aspects of our immune system that we will talk about in detail in this class that contributed to our host defense in, dealing with pathogens. And we separate them into what's called the innate and adaptive immune system. And so what you're going to get in my lectures, since we have to go through so much information so quickly, I'll have simple slides where there's very little information on there that I just use as a prop in my discussion. But then I give you a laundry list. And I realize it's boring and it's not the most, quote, supposed to be the most effective way on teaching and so forth. But I find it's the most effective way to get some of the key things that I think you should take from my lectures in preparing, you know, down the road for applying a knowledge of immunology within the clinic, and also it'll help you on your boards and exams and so forth. So take my lectures as a guide. And so I'm going to give you some boring slides coming up here and so forth. But these particular examples that I pull out of the books and so forth, they simplify it, and we focus on some of the key differences between our innate and adaptive immune system. Our innate immune system is very rapid. In fact, it can be instantaneous. An example of our innate immune system are our physical barriers. So the skin, the epithelial lining of the gut, the epithelium of the lung. We have responses within our innate immune system in which we deliver cells. And you guys are all experts. You know more probably about inflammation than I do from your microanatomy courses and so forth. And then when you take pathology down the road, you certainly will. That the inflammatory response is how we deliver cells to a site of infection to participate in this very rapid response of the innate immune system. We do have specificity within our innate immune system, and by that we have targeting structures that we can target different types of pathogens, but it's pretty limited. Okay, and we'll talk about some of those targeting structures. And once we get the response going, it's about as good as it's going to get. It doesn't improve. It doesn't learn on how to kill the bug. In contrast, our adaptive immune response is slow, but it's incredibly specific. It will generate a response that's needed to get rid of the type of bug that we're exposed to. And it will target on that bug what makes that bug unique. And so it'll be highly specific. And what do I mean by that? If it's a virus that can attach to the epithelial cells of the lung, then one of the targets that our immune system will go after will be the receptor that the virus uses to attach to the epithelial cell. Because if that can get blocked, then you could block the ability of the virus from infecting a whole virus. Brucella isn't going to do that. It doesn't have the same receptor. And so what we target on an influenza virus that will be effective in eliminating that bug from our epithelial tissues of the lung will not work against a Brucella or a Coxiella or even a Vaccinia virus. It will be absolutely specific to the signatures that are unique to that infectious agent. And we're going to spend a lot of time on that specificity because it is the coolest story in genetics of how we can get to that level of specificity. Yeah?
Does the immune system preferentially try and target the earlier parts of the replication process? Or no?
Not unless the molecules of the pathogen will drive that. What will dictate that is how, I'm going to give you a definition here that comes up a little later, how antigenic the proteins are of the virus. What dictates whether or not something is antigenic, meaning our immune system can see it, are a number of factors. One is how different is it than any of our proteins? So how foreign is it? But also size, stability of the protein, the chemical nature. Proteins are very antigenic, carbohydrates aren't. And so it depends upon the molecules that are involved in initial infection, or just an assembly of the virus, for example, and so forth. Depends upon how antigenic they are. So, the adaptive, we have incredible specificity, and then the other cool thing about this is we're going to target what's unique to the bug, and if the first thing we use doesn't work, we're going to improve on it. And so some of us, when we breathe in we use the rhinovirus, so a cold virus. So somebody has already breathed in the cold virus probably when you got back to town after the break or when you were gone on the break. And you're going to mount an immune response to that. Let's assume you've never seen this virus before. So it's a new one. So you're going to go through a classic immune response. Let's say we all get the same virus, and none of us had ever been exposed to it before. So we're all going to go through a classic primary immune response. We will have differences in each and every one of us in the length of time it takes to get rid of the virus. Because my immune system, and some of this is just by chance, it's a statistical issue, my immune system might generate a very, very effective response on the first try and eliminates the bug, and I'm over the virus in a week. Your immune system may not have driven that initial response, and it needs to change, it needs to improve, and that might take 2 weeks for that to occur. And so we see differences in the length of time between individuals and clearance of the bug. And one of the reasons, it's not the only reason, one of the reasons is that we have differences in our initial response And if it doesn't work, we need to improve on that, and it just takes time to do it. The adaptive immune system improves. Within the innate immune system, we have cells. You guys should all be familiar with some of the cells in the innate immune system. Classic cells of the inflammatory response are part of the innate immune system, so neutrophils, macrophages, and they're involved in detecting pathogens. In fact, what's kind of cool with this slide and the terminology, this course next year is going to be called, in the WIMU program, Invaders and Defenders. I have a problem with that, but well, here we have it. We're going to detect an invader, and we have a defender, a neutrophil that will engage that bug. And I guess I have to use these types of terms. It will eat it, won't phagocytize it, it'll eat it, and then kill the invader and so forth. So you guys are lucky, I think, that you're not going to have to be taught this way next year. So we have cells that'll engage, and they have receptors that they use to bind to those bugs, bring them in, and most of the mechanisms is associated with phagocytosis and degradation of the microbe within the phagocyte. Then we have molecules, soluble molecules that are part of our innate immune system that will bind to invaders, and some of them can kill. We've got these small peptides that are called defensins that our epithelial cells produce that have antimicrobial activity. They hit a bug and they can poke a hole in the bug. We also have soluble proteins. We're going to spend a lot of time on some of these that coat the invaders. And so we're going to spend a lot of time on complement. I'm going to talk about it in the context of how our antibodies work, but our pathology, inflammation lectures, which are given by Nicole Meisner, MD in our department, she's going to be talking about complement in exquisite detail. And it's a pretty cool system. Proteins that are normally in an inactive state, that when they get activated by one of three pathways associated with an invader, that we generate fragments of complement, some of which coat the bug, and it makes it easier for our phagocytes to engage and eat the pathogen. And then we also have soluble molecules that can block growth and also limit microbial spread. This will be the only time I've mentioned it because it took years before I even believed it, but I think now the data is pretty good. There's a real cool mechanism of limiting microbial spread that neutrophils engages in that has nothing to do with the live neutrophil. You guys know that neutrophils have a short half-life, right? So when I talk about them, I kind of talk about them as a shotgun. They go into a tissue and they're just going to try to destroy everything in front of them. And hopefully it's a bug that they're dealing with. And however, during the process, a lot of the neutrophils will release their intracellular components. They will lyse, they will die within that tissue. And all of those chemicals inside those granules, and you guys know what's in those granules, it's as bad as household bleach that's inside these granules gets released and damages the tissue. And that's the classic inflammatory response. The reason why the foot swells up, one of the reasons, when you step on the rusty nail, is that the impact of neutrophils that are coming in. However, what's kind of cool is when the neutrophil releases its DNA and so forth, it can generate a net that can encompass the microbes and keep them in that one spot. And so these nuclear nets that neutrophils certainly generate help immobilize the microbe, giving other aspects of our immune system more time to deal with the bug. Our adaptive immune system, now when it's engaged, it will be dealing with either extracellular organisms or intracellular organisms, and it has different mechanisms that we're going to talk about that it uses to deal with them. For extracellular microorganisms, it's the antibody response. For bugs that exist inside our cells, we're going to drive a cellular response against them. We are going to target what makes the pathogen unique and what is different than us. So we're going to target the foreign aspect of the bug. And I gave you guys this term already. This term antigen refers to what is foreign that is expressed by the microorganism that we've been exposed to. And that's what the immune system is targeting. It doesn't inherently know it's a virus. It's targeting the antigens of the virus, and it will generate the appropriate mechanism to eliminate that virus. But the goal of the immune system is to get rid of the antigen. That's why we can develop a vaccine where if we could just take the antigen, not the bug, we could drive the response. We can teach the immune system how to get rid of the bug if we're ever exposed to it. So antigen, this term is critically important to have a good feel for it. The foreign molecular structures recognized by the adaptive immune system are called antigens. Okay? Any foreign substance that we're exposed to that can bind to specific receptors on our B and T cells have the potential to drive that that immune response. And we're going to talk about those receptors in exquisite detail. If it induces response, you will see this other term called immunogen. So we are exposed to a lot of different antigens in our lifetime that are foreign, that we never mount an immune response to. And so there is a difference between multiple antigens and how well they drive an immune response. And so back to the question of, will our immune system preferentially target the early phase of viral entry into a cell? That's dictated by how immunogenic its antigens are, or whether or not they're immunogens that they can drive an immune response. So types of antigens, they can come obviously from any microorganism. It's what defines the microorganism as being unique. The antigens of Yersinia are different than the antigens of influenza, even though they're both viruses. Antigens make up the given microorganisms. They can be proteins, carbohydrates, lipids, nucleic acids are the proteins. But we can drive responses against carbohydrates, lipids, and nucleic acids. But they're never as robust as the response to proteins. I will use proteins as my main example when we're talking about driving immune responses. When we're talking about a microbial antigen, there are lots of different antigens that make up a microbe. There are antigens associated with cell membrane, the cell wall, the pili, the flagella inside the bug. We got lots of molecules we can target. For viruses, it's the same story. They're not as big, they're not as complex, but we got multiple antigens that we can target. Yeah?
Antigen can't be inside the cell unless it...
No, it can't be inside the cell. And this is where those phagocytes that take up the bug and chop it up and so forth, and they're intending to destroy it, they're generating fragments of all the proteins of that bug. Some of those phagocytes, and we'll talk about which ones, can take those proteins and present them.
So it's the
To our- Yeah, they could present them to the T cell by an MHC presentation, or they can just bring protein fragments into a lymph node, for example, that B cells can respond to and
But
so
if
forth.
that protein's intracellular, then it wouldn't happen with the
Yeah, so if we're going to target a protein in here, okay, then that's not going to be effective at preventing this bug from attaching. And actually, it's not going to be effective in a lot of responses unique to the bug. You tend to want to target, for antibodies, for example, these guys out here and prevent attachment in that setting. But there are times when we're going to be targeting, well, that the proteins inside the bug will drive immune responses. And this could just be important in driving T cells. You mentioned MHC. We need to, in most cases, almost all cases, we've got to drive both T and B cells in order to have an effective immune response. The T and B cell do not have to be responding to the same antigen. And so we're going to talk about those examples. And so we could have a T cell that's driven, that provides the necessary help to a B cell to make the B cell work better, but it might recognize an antigen in here, and the B cell recognizes an antigen in here, and we'll talk about how that works in that context. Viruses are very similar, they're just less complex. But it's not simply microbial proteins that can be antigens and so forth. Within the clinical setting, you got lots of examples of other antigens. Anything that's foreign can be potentially an antigen. So, if it's red blood cells that are being used in a transfusion reaction and they're not from an identical twin, they have antigens that are different. And we can target those. In particular, we're concerned about glycoproteins of red blood cells, the blood group antigens. In order to have a successful transfusion, you need to match appropriately the blood group antigens. And we've talked about that when I lectured in the anatomy course, and I'm assuming that probably was talked about in the vet microanatomy course as well. But those are just classic antigens. The reason why you can only accept blood of a certain type is due to the fact that the red blood cells have antigens on their surface that your immune system can target. They simply, in the context of a cell from another animal going in the context of a transplant, I've already given the example of transfusions, but I guess which is a transplant. Unfortunately, and for the ideal situation, we want our immune system targeting non-self. So we refer to that as the foreign aspect of the antigen. That's what we want the immune system targeting. Unfortunately, for you as a clinician, you actually don't get to see that very often because that means everything's working. And so the patient is just fine. It's mounting good immune responses and so forth. They're not sick. You're not seeing them. Or if they are sick, it's not that big a deal. They just have a cold. In order for that system to work, we need to teach our B and T cells the difference between self, all the proteins that make up our body, or an animal's body, and non-self, what's foreign. We've got great mechanisms to do that, but they're not foolproof. And so we do have examples where some of the molecules that make up some of our cells, we do have the potential to drive a B or T cell response. And when that occurs, they're referred to as autoantigens, and we can drive immune responses in those settings and drive autoimmune diseases. That's when you see it as a clinician, either in veterinary medicine or human medicine. We have these examples where we've got B cells that didn't learn the lesson between self and non-self, and they're exposed to a self-antigen at some point later on, and they're reacting normally. Remember what I said? The role of the immune system is to get rid of the antigen. It doesn't really know it's a virus or a bacteria. It doesn't really know that it's a pancreatic cell, that it's mounting an immune response to leading to diabetes, and so forth. It just knows there's an antigen. I got to get rid of it. We have other autoimmune diseases that occur in animals and humans in which the immune system actually targets not a very immunogenic antigen, but when it does occur, it's pretty devastating, and that's when we get an immune response against nucleic acids. And so we can have antibodies against DNA, we can have antibodies against nucleic acids, even the histones and so forth. And so whenever our immune system is exposed to its own DNA, and we can drive an immune response and get antibodies produced against it, and that leads to an autoimmune disease that's called systemic lupus erythematosus, in which we are targeting DNA, our own DNA in that particular setting, plus a bunch of other autoantigens. Then other microbial, non-microbial antigens that are clinically relevant, and if you have food allergies, you know what I'm talking about, but as a clinician, you're going to deal with it. And that some of us will drive immune response against some of the food proteins and so forth. It's kind of amazing that this is a really, really big problem. And Diana over here, a new faculty member in the department, actually works on gastric and intestinal immune responses. And we have this issue that our immune system within our intestinal mucosa and so forth has mechanisms, different mechanisms to deal with food antigens versus, or food proteins versus microbial proteins versus pathogenic microbial proteins that makes the whole system work. And what amazes me is that the mechanisms are pretty elaborate to allow this distinguishing between these different proteins and so forth, that we don't have more problems than what we really do have in the context of food allergies and so forth. But anybody who has asthma and so forth, we have another example where we have non-microbial antigens in which we can drive an immune response that can lead to pathology. And these are what you're going to see. You don't get to see the success, you get to see when it's the immune system's actually contributing to some of the pathology in your patient. So what makes a good antigen? I've already mentioned it earlier. Size, how big it is. If it's too small, a lot of times we just can't see it. The immune system can't see it. How foreign it is. The complexity of the antigen, really the chemical nature of the antigen, protein, lipid, carbohydrate, and stability. I haven't even mentioned that. You know, if it's an incredibly resistant protein to proteolysis and so forth, and hangs around for a long time, that's very immunogenic. If it breaks down right away and so forth, it's not as immunogenic. Yeah. The last term I'd like to get to, and then we'll take a little break here. We talked about an antigen. So let's say it's the attachment protein that influenza uses to bind to our epithelial cells. So it's what makes influenza unique. We certainly don't have it. It's foreign. And let's say it's 60 kilodaltons. So it's appropriate size and it's protein and so forth. Appropriate size, it's pretty big. We're going to target that antigen, but we're going to target different regions. So let's assume this is an example of the influenza attachment protein. So relatively big. We are going to not simply attack the antigen as a single entity, we are going to attack specific regions of the antigen. And these are referred to as epitopes. When I talk about our targeting structures, one an antibody, and then the targeting structure on a T cell, they target these epitopes here. And for simplicity, I didn't talk about the receptor, the antibody molecule, having a mirror image of this epitope. And it's a specific interaction that takes place here. Absolutely, not only specific to the antigen, but to this single spot on this antigen. And that this single spot is different than this spot, and this one's different than this spot. So this complexity gets more and more complex as we talk about what the immune system is targeting. It targets the antigens that are unique to the bug, And then on the antigen are different sites that it targets. Yes?
Does it work almost like an enzyme? You know, like the locking key theory. You know, will different antibodies kind of try out antigens until something fits?
Well, in the opposite direction. And we'll talk, when we talk about the genetics tomorrow, what's cool about our antibody repertoire, just for simplicity talking about it, we've already made the repertoire, okay, of antibodies that could potentially bind to epitopes of any antigen we could theoretically ever be exposed to. We make them before we're ever exposed. And the frequency of those are really low if we have antibodies that are already produced against anything we could theoretically be exposed to, including, and in my previous lectures, I know I've said this, and students that have had me in multiple classes know I say this over and over. I don't care if the proteins ever existed on Earth before. We already have made an antibody against it. So, when we're exposed to the antigen, it is how antigenic or immunogenic that antigen is that'll dictate what antibody response is going to take place. So it's really the antigen that's going to drive that, and it's going to select for the best antibody to drive, and that's going to be dictated by the affinity of the antibody binding to the antigen. That setting. So I don't know if that answered your question directly. We'll be able to talk about that tomorrow in more detail when we talk about this. Yes?
So you said the more complex the antigen, the better, because I'm assuming that means you have more possible epitopes that antibodies can identify. So in a typical immune response, are you usually attacking like the one with the highest affinity, or are you going to be attacking like all of those epitopes at the same time?
You're going to, in most instances, you're going to be attacking multiple epitopes. You may not be attacking every single one equally, because we have these issues on how immunogenic this particular region might be. How immunogenic the antigen is and how immunogenic the particular epitopes are. And the repertoire, again, having to kind of step into tomorrow's lecture, the repertoire of our B or T cells that we've already generated that have the mirror image for these different regions. We've already generated them, but we do have differences in the frequency of those populations for given epitopes and so forth. So you might actually have a decent number of B cells that could recognize these three epitopes, but I might not. I might have more B cells that recognize those epitopes. So we have a statistical aspect that contributes to this as well. But back to your very first point, the complexity of the antigen is to our advantage, that we've got multiple sites that we can target. We rarely will target each and every one of us equally each one of these sites. Did that answer that question?
Yeah. I guess sort of a follow-up question. So if you have a greater population of B or T cells for specific sites, but there is a population that exists, maybe smaller, for a different epitope, if that small population recognizes that antigen, wouldn't it then like ramp up production and create a larger population?
You know, we have those scenarios, and we actually have scenarios where, and this is in the context, if your question is maybe getting to the point of how, as a clinician, you can take advantage of that information, it might be in the context of an appropriate vaccine. So let's say in the context of immune responses against this antigen here, and this antigen in the context of all the other antigens that make up that given bug. We have a concept, we don't spend enough time in this course talking about this, but we have examples with microorganisms that we refer to certain antigens and epitopes as being immunodominant, meaning that most of us have a larger frequency, still 1 in a million, but a larger frequency of B and T cells against certain antigens that dominate the response, but it's not protective. That's a selective advantage to the bug, to actually have antigens that will drive immune responses in nearly all of us if they're non-protective, meaning they don't get rid of the bug. And we have those examples, and certain bugs take advantage of that. So certain parasites have these large glycoproteins and so forth that are very immunodominant, drive immune responses. They're not an advantage to your patient in getting rid of the bug. It may be the epitopes or the antigens for the population of B and T cells that are incredibly low frequency, back to your question that we want to target. And so we probably have examples out there with vaccines that have failed, that we have been driving too many examples of immunodominant antigens responses that are not protective, and we really want to target that low frequency clone of B cells that might be targeting this region here. And that's all in the context of vaccine development that now is taking place. We now know certain antigens, these immunodominant antigens on certain bugs, are not what we want to target. And it's an actual disadvantage for us, and it's an advantage for a bug. And why does that occur? That's just selection. We selected microorganisms in those settings that survived because they had these incredibly antigenic molecules that our immune system was wasting all of its time on. They weren't protective. Okay, I know I got kind of on a tangent there, but did that get to your question? We do have those examples where we want to target that low frequency clone. And we do have examples where people just are driving immune responses, everything is working, but they're targeting the wrong antigen, and we're not clearing infection. You guys have to use antibiotics in those settings. One example where that happens a lot is with strep. We get good immune responses against the strep bacteria and so forth, but you're going to have patients that are going to come back with recurrent strep infections. Maybe some of you guys have had recurrent strep infections and so forth. It doesn't mean you're immunocompromised. You just weren't driving a great immune response against the appropriate antigens for whatever reason in that context. And so, you know, one of my kids had recurrent strep infections. You just have to put them on antibiotics and get it cleared up. And then the bug gets cleared in that setting. That just occurs. So these examples do occur where our immune system will target these immunodominant antigens and it may not be really beneficial. And we really want to figure out how to get at these other cellular responses. So I know a little bit of a tangent. I think I already forewarned you guys before I get off on tangents and so forth. Certain answers. So let's take a break right now, give you a chance to get some water. And so what we're going to do, evidently, I didn't realize this, we're not pressed for time afterwards, so we will take a break midway with this course. I thought we were going to have to do a rush through and so you guys can get back on campus, so it looks like you guys have the necessary time. And we have examples, you have examples down the road as a clinician, unfortunately, that you are going to be using certain drugs. And in most of your patients, the drugs will not drive an immune response, but the drug is foreign. And it's not driving an immune response because it's too small. However, certain things can happen to make this molecule immunogenic. And when it occurs, we classify this type of molecule as a hapten, in which, because of its size, it's not very immunogenic, but it is foreign. But because of its size, it's normally not a problem. However, if the molecule can bind to something that's big, and even if the thing it's binding to is not foreign, it's now on a big thing, and we've overcome the size restriction. And we have multiple examples of this, of molecules that can bind to larger molecules of our own maybe a plasma protein like albumin. And when this occurs, now we can drive an immune response against that hapten. So we use this term in the research setting all the time, but it has huge clinical implications in the context of when this occurs in your patient. And the classic example are with certain antibiotics. So penicillin is the classic example of a hapten that is a foreign molecule that when you're giving it to your patient. But for most of your patients, it's no big deal. Because of its size, you don't drive an immune response against it. However, penicillin can bind to albumin, serum albumin. And when it does that, some of us, relatively small fraction, can recognize that complex. And we drive an immune response against the hapten, the penicillin, in this setting. We sensitize the patient to penicillin, they become allergic to penicillin. And this is not a good thing. So my wife is allergic, dramatically allergic to penicillin. And when she receives a penicillin antibiotic by a clinician that didn't ask, this is what can occur in that we have now driven a pre-existing immune response in that patient due to the prior use of penicillin. Now we come in with penicillin again. It's a small molecule. It's going to go systemic. The patient has driven an immune response, has lots of antibodies against the penicillin because it was able to drive an immune response because of that penicillin binding to the albumin systemic. We get deposition of those antibodies, penicillin, albumin complexes, and so forth, in capillary beds. And it'll present in the skin as an inflammatory lesion. And the reason why we get inflammation is that we don't want to have immune complexes sitting around. We want to get rid of immune complexes. There is a good reason to do that in one setting. Hopefully the immune complex means we have an antibody against a bug. But we got to get rid of it. But we really want to get rid of any immune complex. And the way we do that is we activate those complement proteins I mentioned before. And one of the results of activation of complement is triggering an inflammatory cascade. So in this setting, when the patient has become sensitized to penicillin and they're re-exposed to penicillin, and penicillin is going to go systemic, we can get immune complexes formed and they deposit in all of our capillary beds and we get inflammation. It's self-limiting in this setting, but we get dramatic inflammation depending upon how much of an immune response was driven by the prior sensitization in that setting. Yeah?
What drives the initial immune response, though? Is it that your wife has a different antibody than?
Yeah, so she had, first of all, she had to have a B cell population that could react to the penicillin or the penicillin bound to the albumin. Okay?
So it's not penicillin binding to albumin. That happens all the time. It just depends on if you have a substitute.
There's variation in that between individuals, but there's not a huge level of specificity to that interaction. And so we're going to have it take place. In order to drive an immune response, you've got to have the B cell that can react to it, and it's got to react to it in an appropriate context in order to drive the type of B cell response that can lead to this. In this setting, this is called the type 3 hypersensitivity reaction, and we'll talk about this later on when we talk about hypersensitivity. It also happens in animals. This is not specific to humans. We can drive allergic reactions to some of the drugs that we use, and we see these systemic inflammatory responses that take place. So a little bit of a summary here. Antigens are what we're targeting with our adaptive immune system. The best antigens are large, complex, stable, foreign. Epitopes are what we target on the antigen. And then haptens, you're going to see in the context of your practice down the road, where you're going to have examples where patients have been sensitized to haptens because of the ability of the hapten to bind to some of our cell proteins. So gave you the example of becoming sensitized to a drug like penicillin. But we can also be sensitized to certain toxins, to certain metals, and so forth, by then binding to some of our proteins, so forth. And we're going to be talking about different hypersensitivity reactions. We can have one that's restricted to the skin that's called a type 4 hypersensitivity reaction. And it'll be an example of plant chemicals binding to proteins on our epithelial cells. And we now have an antigenic structure that we drive, unfortunately, in one setting, a cytolytic T cell. And these are our poison ivy, poison oak type responses. Those are haptens binding to our proteins, generating an immunogenic complex. And when we drive CTLs in that setting, that is not a good thing. Because the CTL then, when it sees it, when you're re-exposed to the poison ivy, that CTL is going to see that hapten sitting on the surface of the epithelial cell, and it's going to think that it's a viral infected cell. It's going to lyse it. And that's the lesion that you're going to see in that setting. So, up till now, I've talked about the cool things in the context of adaptive immunity. And again, anybody who's taken a course from me or listened to one of my lectures, I use this slide in every one of my immunology lectures, is that when we talked about innate immunity or adaptive immunity, already I've spent more time on adaptive. All that discussion of what an antigen is and so forth. In every immunology text, you're going to spend more time on adaptive immunity than innate immunity. So you get thinking that, oh, that's the most important aspect of our immune system. It must be the most devastating consequence to a genetic defect. We have a defect in adaptive immunity versus innate immunity. And that's really not the case. And that if we look, and this is just a figure that's representative of multiple different types of microbes, that in the normal scheme of things, we're exposed to a virus, and if you're a healthy individual, the virus will infect, there'll be a level of replication, and over time, we're going to see a plateau and eventual clearance of the microorganism. This is a role of the innate immune system, is to engage early, and its job is really to keep the bug in check, to give our adaptive immune system time to figure out how to kill it. The adaptive immune system can lead to sterilizing immunity, but the innate immune system, its role is really just to kind of keep the initial infection in check. And so this time course here, where we start to see clearance and so forth, it's a week to 10 days. And most of the time, if you've never seen a microorganism before, it'll take a week to 10 days to where, and this is in the good scenario that you're driving an effective adaptive response right away, that will start to clear the bug. So you breathe in the cold virus today, and or your patient gets the cold, and it's going to take a week to 10 days to 2 weeks and so forth to get over it. And that's this normal course. Now, when we look at genetic deficiencies in the human population, or genetic deficiencies in animal populations, or those that we induce experimentally in animal models, either in adaptive or the innate, what we see if we eliminate the adaptive immune system is that we still keep the bugs in check. We just don't get sterilizing immunity. We don't get clearance of it. But early in the course of your patient's life, the lack of adaptive immunity is not incompatible with life. Many times you're going to be dealing with your patient and giving them antimicrobics. And you're going to see then recurrent infections, and we're going to be talking about these molecular mechanisms. I'm going to go through in detail why they're an advantage to you as a future clinician, is that it's going to, if you're you're seeing recurrent infections over and over, the type of infection is going to teach you what aspect of the adaptive immune system is lacking. And then you have potential treatments that you're going to use. Maybe it'll just be antibody replacement therapy because you're lacking an antibody response. My point is, you're going to have time in order to make that diagnosis and so forth. And certain aspects of lacking time to work, even if we had a functioning adaptive immune system, it doesn't engage. And so many times we see the consequence of the lack of an innate immune response as appearing more dramatic. But we don't spend a whole lot of time talking about those mechanisms. We really spend most of the time in this class talking about these, that context. So now we can go through this pretty quick because you guys are experts on cells in the body, cells in the immune system. And so it's going to be cells that are going to be doing everything. Yeah?
Sorry, just on what you were just saying, so if autoimmune diseases, is that usually, that's from the defect in the
No, so an autoimmune disease, this is an interesting dichotomy here. It's not a defect in the immune system in its ability to respond. Innate is working, adaptive is working exactly how they're supposed to. Where it's a defect is your immune system hasn't learned the difference between self and non-self for the particular antigen that's causing the problem. So in lupus, the example I gave, one of the antigens that's a problem are nucleic acids, DNA. Your patient will make antibodies against DNA. So the mechanism that's taking place is perfectly normal. The immune system, the immune cells are doing what they're supposed to do. We just didn't teach the cells the appropriate difference between that DNA being self versus non-self. And that B cell is perceiving that DNA as foreign and is reacting. Everything is working the way it's supposed to. We just didn't get rid of, we didn't teach the immune system not to treat that antigen as foreign.
Isn't that part of the leukocyte development, is to be taught self?
To be taught, and we have genetic deficiencies, and where those lessons are not learned, and it'll present as an autoimmune disease, but the functioning of our immune cells are perfectly normal. They are responding perfectly normally, it's just they're responding to an autoantigen. And so that's different in that context. Those patients still are mounting good responses against viruses, good responses against bacteria. You're not seeing an immunodeficient state in that patient until very, very late, when they have such an overwhelming autoimmune response that it's causing pathology and so forth. But early on, your patient is presenting perfectly normal in the context of responses to microorganisms. So this figure here is purely the response to a microorganism. Your autoimmune patient will be responding to bacteria, viruses, so forth, perfectly fine, unless they've got multiple genetic deficiencies in that setting. Did that answer your question? Okay, good question. Okay, cells. And we use different terms for them, white blood cells, leukocytes, and so forth. We find the players in the blood, but we find them in tissues, and in fact, our immune cells, particularly our lymphocytes, do their initial response in our secondary lymphoid tissues. You should be familiar with, excuse me, the normal frequency of leukocytes in the blood, and not simply to get the question right on the exam. This is an incredibly powerful diagnostic tool for you. When you're going to look at that lab report, when you send down a blood sample from a patient and you're trying to figure out what's going on, these frequencies are very important and these numbers are very important. If you have a septic patient, you have a patient with a systemic bacterial infection and so forth, neutrophils many times are just going out the roof. You see that, it's really going to steer you in where you need to look and what you need to do with your patient, potentially put them on antibiotics and so forth. If you have an increase in monocytes or blast cells and lymphocytes and so forth, you don't suspect a leukemia, and things are not in the realm of a cancer and so forth, this might be indicative of something like mononucleosis, in which simply the changes in these frequencies are going to start to get you thinking of what could be taking place in the patient. And so there is a value, there really is a value to keep in mind the frequencies of these populations of cells. And if any of you have chronic asthma and so forth, and I took a sample of your blood, you'd have a lot of eosinophils. You know, they're the bane of your existence in context of the response that's taking place in your lung when you're breathing in an allergen. And you guys should be familiar with the morphological characteristics of these cells. Yeah?
Why are the ratios reversed in kids?
Well, that's an interesting thing. And we have differences in different animal species and so forth. But our immune system needs to develop after birth. And we have continued development that takes place. And unfortunately in this course, and I was going to add it this year, and I decided I've already added too many new things and so forth, but our flora, our normal flora, the microbiome, you guys have all read about this. You guys will be right at the forefront of whether or not you want to advise people to take probiotics, or maybe you'll have defined microbes that you're going to give patients and so forth. That has a huge impact on the development of our immune system after birth and so forth. And so when we're responding to our normal flora, we're responding to other developmental factors and so forth, we will drive a reversal of the populations within the blood, and it's just responses to those external factors that are coming. Some of them can be microbial products. Some of them are responses of other cells, like tissue cells, to microbial products, and it's an indirect effect, and so forth. And we will start to see a reversal. We'll see a larger number of neutrophils in that setting. Define morphological criteria for each one of these populations. And again, you should be familiar. You should be able to look at a slide and say what's a neutrophil versus a lymphocyte versus a monocyte, and so forth. We're going to be talking about a technique we use in the research lab. You're going to rely on it in the diagnostic lab for looking at these populations very quantitatively and rapidly, and that's flow cytometry, in which a machine's going to give you the characteristics of these cells, and you need to interpret that. You know, one of the reasons why I'll spend a little bit of time on this is that do not always assume that the diagnostic lab is right. They do make mistakes. Anytime a human's involved in anything, there's potential for mistakes. And so your role as a clinician is to provide another level of assurance, and you've got to look at that data to see if it makes sense. And understanding and knowing these populations, their frequencies, and their appearance, and so forth, will be another level of assurance that the data you're getting on your patient is real, that will help dictate what you're going to do as far as a treatment. You should also be aware and refer back to your previous lectures and so forth where these cells come from. So we have the hematopoietic stem cell, the bone marrow after birth, that's giving rise to our populations of immune cells that are doing everything, in which we have progenitors that lead to B cells, T cells. We have progenitors that lead to our myeloid cells, neutrophils and eosinophils. Monocytes, when monocytes get into tissues, we refer to them as macrophages. This is overly simplistic that monocytes lead to dendritic cells. We have dendritic cells. That's still kind of a black box in some settings, and I'm going to rely on Diane to correct me when I'm overly simplistic on dendritic cells. But these guys are all contributing to important responses within the immune system. And, you know, our stem cells are giving rise to our platelets and red blood cells. But again, you guys are experts on that. So now we've got a little bit of historical perspective on understanding the immune system, how it's an advantage to developing vaccines, examples of vaccines, what's an antigen, and so forth. So let's go through very quickly examples of responses against these pathogens. Let's bring you through the process here. So we need to break through our innate defenses if the bug is going to cause disease. We have a very, I mean, you think of it as kind of simplistic, but a barrier that has to be overcome, that epithelial barrier. Be it in the lung, be it in the gut, be it in the skin, and so forth. But we have some pretty good pre-existing host defense mechanisms that make it tough for a microbe to gain access into the underlying tissue in order to cause disease. One of the things we take advantage of is our flora. That it's really important to have that microbiome in our gut because they are contributing to a host defense mechanism. They make it tougher for a lot of microbes to gain access here. Some of it's as simple as competition. And in that the salmonella that you eat and so forth is not having as easy of a time to get to the epithelium because of this flora. Unless your patient is inappropriately using antibiotics. And this is a big problem. You know, the literature is filled, both the popular press and the scientific literature, of the negatives of overuse of antibiotics in the context of driving resistance in microbes to the antibiotics. Huge problem for you as a clinician. But there's another big problem. When you use antibiotics, you knock down this normal flora. We lose the competitive advantage here. And Ivanka and whoever will be teaching bacteriology in the vet program will be talking about what happens with overuse of antibiotics, in which it allows certain bugs to gain a toehold in humans. C. diff, Clostridium difficile, big problem for you. And that use antibiotics, boy, in a certain fraction of your patients, that's a nasty, nasty bug. That is now competing with MRSA. Right, Dan? You know that? I think somebody said that just to argue with Ivanka. That competing as far as frequency of infection in hospitals.
It's kind of interesting in that context. There's a new treatment kind of that's coming to the forefront for the gastroenterologist to treat C. diff, and that's by putting back those microbes by fecal microbial transplant. You may have heard of that.
Yeah,
so it's pretty massive. It works pretty well.
Yeah, you know, in Europe they tend to jump in doing those things a little quicker than in the U.S. and there's some good data in the clinical realm for that. That still scares the hell out of me. And that you know, I'm a believer in the role of these guys, and I think the real big role is how they help regulate our immune system and so forth. And when you get rid of them or you alter it, you have these deleterious consequences. And we have these examples where you can go in and replace, and it's a great treatment. You see this tremendous response, beneficial response in your patient. However, we are a long ways from having a handle on that community there. It's complex, and to think that we know enough that we can do that consistently, particularly in the context of the use of probiotics, is we're a long ways from that. It doesn't say that there aren't these dramatic positive benefits and so forth, but don't assume it's just going to be this end all for all your patients. It depends upon my microbiome in Bozeman, Montana, and so forth. If all of a sudden I'm given fecal sources from another region and so forth, it may raise havoc just like it did in Mexico before Christmas for me. That microbiome raised havoc. But others in my party that ate and drank the same food, it didn't cause any problems. And so we all are going to respond differently. But that's my soapbox on that. Yeah.
Do antibiotics and feed have a significant reduction?
Antibiotics, anytime they're going to be used, can do this sort of thing. And you still, this is not to downplay the impact on resistance. And certainly the overuse of antibiotics in food animals, it's a concern in generating resistant microbes. Just imagine, You know, you got this problem of E. coli in our hamburger and so forth. And if we're going to use antibiotics to enhance growth and so forth, and we've selected now for certain antibiotic-resistant E. coli, it's going to be carried through the processing. It's going to be carried. It's not going to be a huge problem for the animal, but then we're at the Jack in the Box eating E. coli hamburger, and it's a problem in that setting. And so we've got that issue. But here, we're gaining a greater and greater appreciation of this negative effect of overuse of antibiotics. And so the book doesn't, we just don't spend a lot of time on it, but you as a clinician, you're going to be right at the forefront. Just like Diane said, it's in the clinic. They're doing this, and your patients are going to be taking probiotics. And give them a little dose of reality. They don't know what they're taking in there. That's been sitting on the counter for X number of years. There's already been examples. There are beneficial bacteria in some of those probiotic preps, but they change over time. And to assume what's in this vial 20 years later from the original isolate is the same is really naive, and it's a big problem out there. All right, so get back off that tangent here. All right, so let's use this example where a pathogenic bacteria gains access. Now it's going to break through the physical barriers, and it's going to engage. And the physical barriers also have other very simplistic aspects of our host defense that contribute. There is a chemical contribution, and so secretions in certain tissues contribute to a host defense. Within the mouth, our saliva has some antimicrobial activity, and wouldn't think so every morning, but it does. There are chemicals that contribute. We have mechanical effects. There's a real advantage to coughing, to sneezing, to tearing, diarrhea. It's a physical expulsion. And we actually have mechanisms that trigger those, because that physical expulsion actually can be beneficial. And one aspect of our immune system that we drive that leads to that is an IgE antibody response that's linked to mast cells. And it leads to physical expulsion of whatever drove that. And historically, those were things like parasites that would drive response, helminths, worms in the gut and so forth. How are you going to get rid of that? Physical expulsion can be pretty effective. How do you get rid of what gets into your lung that might drive that? Sneezing it out. Well, you as a clinician, at least in the States, most of the time you're going to see that that's the mechanism that drives allergy responses. And it's physical, but it actually can lead to contributions of reducing microbial spread and so forth, and we talked about the flora. So we break into the underlying tissue, we're going to engage our phagocytes. And so we have our defender here, we have our invader, and we're going to take it up and we're going to eat it in the context of the phagocytes that either are resident in the tissue, macrophages, or recruited to the tissue. Those are predominantly neutrophils. And we have receptors on our phagocytes that recognize structures that are unique to classes of pathogens. Not individual bugs, but classes. So we have receptors that are specific to gram-negative bugs, gram-negative bacteria. We have receptors that are specific to gram-positive. We have receptors that are specific to viral nucleic acids. We refer to those microbial molecules as patterns, and these receptors as pattern recognition receptors. And so pathogens have these pathogen-associated patterns, molecular patterns, and just notice molecular is not there. And we have receptors for those patterns. And a class that you're going to learn about, a class of receptors, are referred to as toll-like receptors that recognize different microbial patterns. They might be gram-negative LPS, might be structures within peptidoglycan from gram-positives, might be viral nucleic acids. And we have a lot of these tolls. I think we have 11 that have been defined that recognize different patterns that contribute to a macrophage taking up the bug or a macrophage recognizing the bug within the cytosol. And that allows the bug, well, the macrophage to engage, hopefully eat, but if it doesn't do the job, it calls for help, right? And we drive a neutrophil response, an inflammatory response, and the bone marrow can be mobilized to put a lot of neutrophils out into the blood that are recruited to that inflammatory site, and we get the classic inflammatory response. So we have an insult, the resident cell tries to deal with it, and when it's engaging the bug, it actually gives out signals that in the vascular system, the blood system, where our neutrophils are flowing, we will recruit cells to this site, and it's a very active response in which the endothelium plays a very important role. The endothelium actually responds to many of the factors that are being produced, and they become sticky. And the leukocyte that is normally flowing within the blood recognizes that. It rolls, it tightly adheres, and then it migrates into the site to contribute to this chewing up process here. That's the classic characteristic of an inflammatory response that Nicole will go into detail on. The mediators that trigger this, the types of cellular responses, the pathology associated with the fluid phase versus the cellular phase, and the types of drugs you have available in dealing with all of this. So we have cells going in, and I told you we have soluble proteins, and we'll talk about soluble proteins that contribute to dealing with the microbe. Some of these proteins are simply intended to coat the microbe and make it easier for our phagocytes to take it up. So in addition to those PAMP receptors, our phagocytes have receptors for these proteins that will coat the microorganism. So a prevailing theme when we're talking about immune responses is that redundancy is the common feature of most of our immune responses. We rarely rely on a single receptor ligand interaction. If one works, two are going to work better. And we will see that over and over again. So the best way for a phagocyte to deal with the bacteria is to have a toll receptor that can see the bacteria, but also maybe receptors for complement proteins that are coating the bacteria that facilitate uptake in the eating step. All right, here, beginning of my bullets, laundry list, that really should be review for you from microanatomy or histology that you guys took in the fall. So if we break through those innate defenses, and I made that really simple, right? Only 2 or 3 slides, but really important. If we break through, we're going to engage our adaptive responses. We're going to engage lymphocytes, B and T cells, that we'll spend most of our time talking about. B and T cells don't do their work in a vacuum. They do it in defined tissues, and they are generated in defined tissues of the lymphoid system. And we have 2 classes, primary and secondary. Primary lymphoid organs are the site where B and T cells are generated. They develop, they don't necessarily have to finish development in those sites, but it's where they're generated. The majority, but not all, the majority of the lesson for self, non-self discrimination occurs in the primary lymphoid organ. Secondary lymphoid organs are sites where the B and T cell is going to initially respond to antigen. So the lymphoid organ system is providing a tissue site for the B and T cells to respond to antigen, and the nature of the lymphatic system, the efferent and afferent lymphatics, provide a conduit of collection of things that are foreign, concentration in a draining lymph node, for example, and then the site for B and T cells to come in via the vascular system and potentially find an antigen that they can react to. And so, in the context of our cells, B cells develop in the bone marrow in humans, primary lymphoid organs, and are initiated in the bone marrow of all animals, but we see animal specificity in the context of final development. I think there's even textbooks that are written by MDs that say B is for bone marrow. That is not the case. B is for bursa, and it was in chickens that this was first shown. That's where the B came from. And T cells, on the other hand, develop in the thymus, and that's where the T came from, the thymus. B cells responsible for antibodies. T cells are cell-mediated response, and this is just a simplistic cartoon, the distribution of our primary and secondary lymphoid organs. We have a recirculatory loop between our blood system and our lymphatic system, which provides lymphocytes that are in the blood access to all of our secondary lymphoid organs. You guys know there's quite a few of them. We've got a number of lymph nodes, mesenteric lymph nodes, the spleen's a secondary organ, the Peyer's patch, throughout the body. Any of those sites could be the site that antigen is collected into. B and T cells don't inherently know where antigen's coming in, and so they gain access to all of our sites, our secondary lymphoid organs, via the blood. And they look for antigen within that secondary lymphoid organ, and if they don't find it, then via the lymphatics they'll enter back out in the vascular system again. Now this isn't absolutely true in all animals, and in fact, pigs are weird, right, Diane?
Well, they have inverted lymph nodes.
Yeah, it's really weird. And that is relevant not only for veterinary medicine, but for human medicine, because designer pigs are one of the things that we're looking at for transplants. That we can go in and clone pigs, and we can put antigens in, we can put in a human MHC and so forth. So we may be transplanting pig organs, but it's really weird, right? Their lymphatics go backwards.
Somehow.
Yeah. And this is just some bullets in the context of this process. You guys also know that within our secondary lymphoid organs, it's not just simply a bag they're going into. There are defined regions, and it's within these regions that we will get our T and B cell accumulations. And for simplicity here, we look at a cross-section of a lymph node in this cartoon. The blue depicts T cell zone, yellow B cell zone. These are referred to as follicles. These are referred to as the cortex of the lymph node. And then later on, though I guess we got a picture here, if the B cells are getting activated, meaning they're seeing antigen, then we get a region within the follicle that becomes less dense, at least in the context of histological stains. And this is due to the fact that these are B cells that are proliferating. They've converted to plasma cells. We're getting some antibody generation. They have a lot of cytoplasm, and that's referred to as a germinal center. And that's an activated follicle that that's taking place in. If your patient lives in a bubble and never is exposed to a single antigen in their lifetime, they won't generate germinal centers. And so we can do that in an animal system. And in the few examples where humans been in bubbles because they lack certain aspects of their immune system, you don't get antigen stimulation and you don't get germinal center formation. Bullets, you guys can just read these at your leisure. In the context of what's going to be taking place, we're getting B and T cell responses. B cells will generate plasma cells that'll make antibody. We're going to generate T cells. They could be a helper or a cytotoxic T cell that we're going to talk about. We've already talked a little bit about frequency of our B and T cell populations that can react to signatures that are unique to pathogens. Frequency really low. It's simply a numbers game. The bug is winning at the start, and we need to increase the frequency. of our antigen-specific cells. So proliferation is the rule of the day for an effective response, and that will increase the size of our secondary lymphoid organs. So when the neck hurts due to the cold virus that you breathe in, that's a good thing. Things are working and so forth. And this is just a schematic of the process. You step on a rusty nail, I've always used as an example. We introduce bugs into the tissue. The phagocytes take it up, driving responses. We bring neutrophils in. Some of the bug, free bug, as well as pieces of the bug are brought in by cells within these sites. Some of them are macrophages, some of them are dendritic cells. You guys know about Langerhans cells that are out in the skin that can take up, highly phagocytic, take up these things. And what they're doing is, if they have broken through here, let's deliver the signatures to the draining node to provide the access for our B and T cells to respond. Same thing occurs in the spleen, but it's in the context of responses to blood antigens. We have compartments within the spleen, B and T cell zones, and again, just refer back to your histology lectures and so forth. In the gut, our largest lymphoid organ in the context of the surface area that it has to respond to, we have these regions that are called Peyer's patches that are made up of T and B cell zones. But we don't have lymphatics delivering antigens to the site. We're getting antigens directly from the lumen of the gut. And we have a very specialized epithelial cell called an M cell that has a very important role. And that is to provide a level of selectivity in the delivery of microbes and microbia in the underlying Peyer's patch. That we'll preferentially, it's not absolute, get salmonella delivered or salmonella antigens to this region versus food antigens that you may be taking in at this site. These cells are critically important. We're getting a pretty good handle on targeting of the M cell, and I think a new class of vaccines down the road that you guys will get to use will be vaccines that selectively target the M cell. So we get the antigen we want right to the M cell, to the mucosal tissue. Because this particular lymphoid tissue does react differently than a peripheral site. And one of the reasons is this difference in how it has to react to food antigens versus normal flora antigens versus pathogenic microbial antigens. There's a lot of unique control mechanisms going on in the gut that don't necessarily take place out in the periphery. And we probably have had a lot of vaccines, not a lot, but we've had vaccines fail to provide protection at the intestinal mucosa because we went into the periphery with the vaccine, with a shot, instead of really driving the appropriate response within the gut. And so there's lots of attempts to develop new vaccines using the same vaccine antigens that are currently out there and get them delivered appropriately to this site and drive the type of response that we want. Yeah?
How do they make it through the acidic environment?
What's that, the antigens? Well, that's in the context of responses with the intestinal tract. The microbe, if we're talking about the normal response, and we're going to deal with the microbe's going to get through. Some of them will die in the stomach. But obviously salmonella is going to make it into the intestinal tract, down to the colon and so forth. And then we'll get driving of responses. If you're talking about the vaccines that are being driven, there's different approaches. One approach is to use salmonella. In fact, the new typhoid vaccine, instead of having a peripheral injection, you have an option of taking the oral typhoid vaccine. Anybody take that? Yeah, I didn't do it with South America, and I took the oral typhoid. That actually is a live, it's attenuated, it's a live salmonella bacteria that you're taking. That'll make it through the stomach. That'll make it into the intestinal tract, and it'll make it to the M cell. And we will drive an immune response. It's just not going to lead to disease because it's attenuated in that setting. We'll talk about that type of vaccine. So in that case, we use the bug to do it. There are other ways where you use the packaging that can make it through the low pH of the stomach and they get release of contents, with the intestinal tract. But Diane actually looks at antigen uptake and responses within the stomach itself, and it's something in immunology we tended to ignore. And there are dendritic cells that take up antigens and so forth, from the stomach itself. But there are multiple approaches to be able to make it through that step. So we'll end with this. Yeah, clear back there. I'm a little over, but you guys have lots of time. So we'll end with this in the context. I still have a few other slides we got to get through before we talk about B-cell genetics and so forth. But you guys don't get to see when everything's working, right? You're not going to see your patient when the immune system is doing its job and so forth, unless it's doing too good of a job. And you might have to go in and use approaches to shut it down and so forth. One setting where you will see it in your patients is when it fails to do its job. And we have a number of reasons why our immune system will fail to do its job. The number one reason in the world, not necessarily in the US, for example, in the world, is actually malnutrition. And that a poor diet impacts then the energy status of our cells, it impacts our bone marrow production of our immune cells and so forth, and leads to an immunodeficient state. However, in addition to malnutrition, we can have genetic deficiencies. You will see a patient that will have a genetic deficiency in some aspect of their immune system. It might be a gross deficiency where both B and T cells are affected. We'll talk about those examples we have. We have them in animals, we have them in humans. And we know the molecular mechanism behind those genetic deficiencies. You have selective deficiencies. Those are going to be tough to diagnose right away in some settings. And having some insight into what is being presented in your patient, what the mechanism behind that presentation is, will help you lead into that diagnosis. We have drug-induced. Some of you are going to do it on purpose. If you become a transplantation surgeon, your goal is to shut down the immune system so that heart's going to take in the patient. You're going to be going in and shutting down the immune system. But you also are going to do that in autoimmune diseases. You're going to go in and try to shut down the immune system. And unfortunately, the most effective drugs still today are the sledgehammers, the corticosteroids, the cytotoxic drug, methotrexate, and so forth, where you're just hammering the immune system. We're coming up with more selective approaches, but we still hammer the immune system in some settings, and those lead to an induced immunosuppressed state. And then we have pathogen-induced deficiencies. We'll spend a little time in the WAMI specific, of course, talking about AIDS, but we have that in animals as well, where pathogens, we have feline leukemia virus, that induces suppression. CMV induces suppression in your patient. Measles virus can induce suppression, and that's what you're going to deal with as a clinician. So we'll end with this at this point. Start up tomorrow with just introducing our antigen receptors and then move to B-cell genetics. Yes?
On that previous slide, what was this, the map? It says total cases or new cases of deaths, or cases of what?
That's a good question right now that I'm blanking.
It must be HIV cases.
The blue, what's the difference between blue and red though, or blue and pink? Cases and death. You can see it down here? Okay, cases and death.
HIV infection.
Yeah, sorry, but I did not remember the blue and
HIV
pink.
infection, not AIDS. Well, yes. AIDS.